Hi everyone, my name is Malvika. I'm an application scientist at Elucidata, and I'll be talking to you today about transforming precision medicine with data-centric approaches. So, a brief introduction to precision medicine. The goal of precision medicine is to prescribe the right dose of the right drug to the right patient at the right time, and it takes into account variations in genes, environments, and lifestyles between patients to personalize medical decisions. So, a very simple example to show precision medicine in action. Let's take the example of warfarin, which is an anticoagulant, and it's prescribed to cardiovascular patients who are at risk of developing a stroke. So, using the conventional treatment strategy of one size fits all or one dose fits all is not possible in the case of warfarin because it is metabolized very differently by different people. So, this precision medicine approach, known as genotype guided dosing, which takes into account genetic information in addition to clinical information on a case by case basis, has shown very high levels of success in clinical trials and is more likely to be successful because it takes into account individual patient information as opposed to one dose fits all. So with the advent of high throughput technologies such as omics technologies, there's been an explosion of uh, biomedical data, and these data sets are typically large multidimensional data sets. So researchers are increasingly turned to ML models to help make sense of this data, analyze and identify patterns in it. And it has seen numerous applications in precision medicine, specifically useful for diseases that show high levels of heterogeneity, such as cancer. So when it comes to precision medicine applications in cancer of machine learning models, you'll find it being used in diagnosis for identifying novel molecular subtypes of diseases, for drug repurposing, in case of combination therapy for identifying the best or most suited uh, drug combination on an individual patient basis, for patient stratification, identifying which patients will respond to a certain drug and which won't, and to also for predicting survivability and prognosis outcomes. But there are certain challenges in translating precision medicine research to the clinical setting. Mainly this lies due to the fact that this is due to the fact that cancer cell lines do not accurately recapitulate tumor heterogeneity, and a majority of ML models are only trained on cell line data owing to the lack of availability of patient data. And so, because of the significant divergence of patient samples of cell lines from patient samples at the molecular level, at the phenotypic level, at the genetic level, it is very hard to translate these findings from a preclinical setting to patient contexts. As a recent study showed, around 50% of hepatocellular carcinoma cell lines taken from CCLE do not even resemble their primary tumors. And this arises to a large number of factors. There are changes introduced owing to the cell line being in culture for a long period of time and repeated propagation. Then there are genomic alterations that arise spontaneously. So to put it very simply, our assumption is that as researchers, that cell line tumor resemblance is very realistic and it looks like this half of the image on the left, but in reality, it is probably more like this comical distorted version on the right. So I'll be talking to you about data-centric strategies that you can use to improve translational outcomes of your preclinical research. And I'll specifically be touching upon selecting appropriate data and selecting features. But before that, what do I mean by data centricity? Data-centric strategies focus on enhancing the data itself to improve the accuracy of ML models rather than reiterating upon the ML model and the underlying algorithm. So I'll be touching upon selecting appropriate data and selecting appropriate features. I'll be talking about the importance of the model system, the type of data used, and the importance of data quality as well. So let's start with selecting appropriate data. As I mentioned earlier, the issue is that the prevailing issue is that a lot of ML models are trained on cancer cell lines, owing to the fact that there's a dearth of appropriate patient samples available for training the models, and this is a problem because tumor like cancer cell lines are unable to accurately recapitulate a lot of the phenotypic and molecular characteristics of tumors. So there are novel model systems such as patient-derived xenografts and 3D organoids that have been developed, and they are sort of known to more closely resemble native tumors as compared to cancer cell lines. But cancer cell lines do have their advantages. They are the reason that they are so widely used today is because they are so easy to procure. There are multiple cancer cell lines available for each type of cancer. It's very easy to conduct high throughput drug screens on them, owing to the large availability of biological material. They're very easy to maintain, store, use, and propagate in culture. But again, the trade-off is that they're pretty divergent at the molecular and phenotypic level from the actual tumor itself. So it's very difficult to translate these findings to a patient setting. So what are organoids and PDXs? Organoids, 3D organoids, they are actually derived from the patient tissue or from embryonic or pluripotent stem cells by administering physical and biochemical cues, and they have organ level functionality. PDXs, on the other hand, are actual patient tumors that are taken from the patient and surgically implanted into mice. They are extensively used to develop new drugs and for testing the efficacy of existing therapeutics. So, reason that organoids and PDXs are known to be more effective than cancer cell lines is because they are very representative of the heterogeneity of the cancer itself, of the tumor, and they tend to retain as well a large number of the phenotypic and molecular characteristics of the native tumor, which is very important when you are trying to translate findings from preclinical to clinical. So this particular paper uses and describes an ML model that was trained on organoid-derived data and was able to accurately predict patient responses in a clinical setting. So one important goal of precision medicine, as I mentioned earlier, is to stratify patients on the basis of which patients will respond to a certain treatment and which patients won't. So this particular paper deals with the issue of patient stratification. They use pharmacogenomics data derived from 3D organoids and trained ML models, which would then predict biomarkers. And the biomarkers were further validated in patient data and were able to accurately predict which patients would respond to a drug and which would not in terms of survivability. And this was done in two different contexts for 5-fluorouracil in colorectal cancer and for cisplatin in bladder cancer. And in both cases, they found that the biomarkers they identified using the ML models trained on organoid data was very predictive had a very high predictive capability of patient response and survivability in both these contexts. So basically, this shows that organoids and other novel model systems that more closely resemble native tumors have improved or potentially have better translational outcomes as compared to the existing status quo of using cancer cell line models. And this is an aspect that as researchers we should keep in mind while training ML models. So the next aspect moving beyond the model system is the type of data that you use. So as we know, there is a whole plethora of different data types that exist today with high throughput sequencing and high throughput technologies, and there are upcoming ones that I haven't really mentioned here, such as spatial transcriptomics. So the very common ones that we use or are aware of are genomics, proteomics, metabolomics, RNA-seq, single-cell RNA-seq, methylation, etc. The common paradigm for cancer treatment so far has relied on identifying specific oncogenic mutations in tumors in the cancer genome using genomics data and using these cellular vulnerabilities to devise therapeutic decisions and strategies. But the success of this approach has been limited to a very small proportion of patients. And so now researchers are looking beyond just using genomics and using other types of data or other combinations of data types to improve the odds of translational success. I'll again illustrate this with a couple of examples from literature. So this particular paper compared genomic and expression features for cell lines taken from the GDSE dataset from the GDSE repository, and they used it to predict cancer cell viability. So basically, they compared genomic features such as copy number, mutation, methylation, etc., and along with the expression level of genes. So they found that when ML models were trained on expression features, they performed better in identifying cancer cell viability as compared to ML models that were trained purely on genomic features. So in this other example, basically they predicted in hepatocellular carcinoma patients, ML models were trained on multiomics data. They used 3 different data types, microRNA sequencing, RNA sequencing, and methylation data. And the ML model then was able to predict 2 subcategories of patients, one with better survival outcomes than the other. And this was validated or tested in 6 independent cohorts and showed high levels of translational success. So basically the point I'm making here is that the data type that you use to train your model has a very high predictive power of the translational outcomes of that model. And so this is not to say that one data type uniformly performs better than the other. The point here is that you have to take into consideration your biological use case and context and evaluate different data types available and pick one or a combination of data types that is most likely to have a positive translational outcome. Another aspect with respect to data is the data quality itself. Now, this is the most obvious aspect. Everybody knows that the quality of data used for your training ML model directly affects the model performance. But there are some typical data quality issues that are very widespread and very common that people are aware of, such as presence of noise, outliers, having missing or duplicate data that is biased or not accurately representative. So, and there are numerous ways that in which you can address for it by using, for example, in the case of missing values, there are different methods by which you can impute or address the missing values, especially in the case of multi-omics data, where you can't proceed to the actual downstream analysis if you have missing values. But what I would like to specifically talk about is other aspects, other confounding factors, which are not as well known or as well discussed in the community, such as tumor purity. So what I mean by tumor purity here, especially in the context of cancer research, this is very important as a confounding factor. And tumor purity refers to the actual number of cancer cells that are present in a patient sample, excluding all of the other intervening cells such as stromal cells, immune cells, etc. So some recent emerging lines of evidence have identified tumor purity as a confounding factor that is not often accounted for and could potentially be a factor that impedes translational translatability of preclinical research. So I'll illustrate this with an example from literature again. So for this particular study by Siroto et al. In 2019, they examined the role of tumor purity as a confounding factor in translational research. And this particular figure, they've compared data from patient tumor data from TCGA with cancer cell line data from CCLE. And they found that the correlation between high purity tumors and cell lines is higher than the correlation between low purity tumors and cell line and their corresponding cell lines for 15 out of 20 cases. And this makes a compelling case for the fact that tumor purity does have a role to play in when it comes to translation. And further, another analysis they did was gene set enrichment on the differential expression results, and they found that the major biological signal for primary tumors, which was absent in cancer cell lines, was upregulation of immune responses, and this they say is possibly due to infiltration by immune cells at the site of the primary tumor itself, and this is something that is often not accounted for. So another thing, in addition to the common quality issues that have to be considered while training an ML model, in terms of precision oncology specifically, is to take into account other unknown factors or confounding factors such as tumor purity, which could also play a role in affecting translational outcomes. So far, I've discussed the different aspects of data that you should take into consideration while training your ML model, such as the quality, the source of the data or the model system, and the type of data that you would use or combination of data types you would use to train your model. Now I'll talk a bit about selecting the appropriate features as inputs for your ML model. So one predominant problem that has prevailed in precision medicine is that identifying high quality biomarkers that are accurate and robust as well as reproducible across different experimental conditions and platforms. And one way that you can sort of mitigate or control for this is to select the appropriate features. What does feature selection do? Why is it important? It helps in reducing noise and redundancy of high dimensional biomedical data. It helps reduce the complexity of biological systems and helps improve the interpretability and translation of ML models. So I'll talk about one particular example. So in this particular paper, the authors sought to compare different sets of features either individual gene expression or pathway activity levels and see if they maintain which were more reliable and maintain consistency across different platforms and different experimental setups. So in this figure in A, they compared data sets, a data set taken from GDSC and CCLE data sets using both single gene and pathway activity level contributions. And so the distances between the two data sets here is lower for pathway activity level in blue as compared to uh, the single gene contributions in red, indicating that pathway activity levels are able to account for experimental variations and show high level of consistency. In B, a similar approach was taken for ensuring consistency or checking for consistency across platforms. And what they found here was that for RNA-seq data and microarray data, the distances were shorter, indicating higher degree of similarity for pathway activity levels and compared to single gene expression, which showed higher distances or longer distances and lower levels of similarity. So basically, using pathway activity levels is able to account for experimental variations and platform variations across experiments as compared to gene expression, individual gene expression. And so another question they asked was, are pathway activity levels also better predictors of tumor subtypes, and are they able to distinguish between tumor and normal samples as well? So for this, they use the Tisney dimensionality reduction method. So figure C is basically corresponds to individual gene expression. And so while the samples were able to partition accurately based on the tumor subtype, they weren't able to distinguish between normal and disease samples. Whereas in the pathway activity level models, you were able to see that there was a better separation both in terms of the actual tumor subtype as well as between diseased and normal samples. So basically, this study sort of makes a case for the fact that pathway activity levels can be used as input features since they better account for differences in experiments and platforms in comparison to individual or single gene expression features. So with that, I've come to the end of my talk, and the key takeaways are that data-centric ML strategies focus on improving the data rather than the actual model, and these can be applied to precision medicine to bridge the gap between preclinical and clinical context. So we spoke about two main aspects: selecting appropriate data and selecting appropriate features. With data, what we have to keep in mind is to select the most appropriate model system, choose the most suited data type or combination of data types for your application, keeping in mind the biological use cases and contexts, and using the most high quality data and while accounting for confounding factors. And you also, for selecting features, you need to keep in mind that you choose features that are able to account for variability across platforms and experimental setups, and thereby reducing the complexity of biological systems so that the interpretability and translatability of the ML model is improved. So with that, I'll wrap up and I thank you all for watching.